Swayam Prabha. Digital India. Educated India.
Hello everyone, we will continue on the course on computer aided drug design. We will talk about some of the issues in the drug discovery process. So let us recap. Drug discovery is a process by which a drug candidate is identified, okay, and partially validated for the treatment of a specific disease. So as I said, partially validated because it becomes a drug only after it gets approval from FDA, that is Food and Drug Administration of USA. So in order to get approval you need to go through all the various preclinical trials involving animals and then human volunteer trials. So until then it is called a lead molecule. So in my lab I may get a nice candidate which looks very promising, which shows very good activity in my activity screen, maybe anti-inflammatory activity or anti-cancer activity. So I call that as a hit molecule, and then I go and finally zero in on one candidate. I call it as a lead molecule. The lead molecule is what is being tested in the preclinical clinical trials, and these lead molecules should have not only the activity but other properties which we are going to spend a lot of time on. So the drug discovery, we need to understand the mechanism of action, we need to identify the target, which target or enzyme or protein it is going to go and bind to and inhibit it or inactivate it. We need to validate that target, and then we need to optimize the lead. So there could be one possible candidate which looks very promising, so we may have to change some of its properties so that it increases solubility, maybe the toxicity is reduced, side effects is reduced. That is what is called lead optimization, and it should satisfy all the ADME properties. Okay, yesterday I mentioned A is absorption, D is distribution, M is metabolism, and E is excretion. So it should get absorbed very nicely into the human system, okay, absorb as we can say, and then comes distribution into the bloodstream, okay, maybe it may get distributed into the various tissues, okay, then it should get it may be getting metabolized, and then finally once it has done its job it should get excreted. That is also very important. So it should satisfy these ADME properties, and then it should have good pharmacokinetics and pharmacodynamic property. Okay, yesterday I did talk about pharmacokinetics and pharmacodynamics. Okay, so all these and then of course it should not have any toxicity. All these are very important. So the drug discovery, we are not going to talk about preclinical studies, clinical trials, regulatory approval, sales, marketing and so on actually. Okay, these are not part of the drug discovery process. Okay, so it is a very long process, it involves a lot of money. As you can see here, in silico, when we say in silico we use computational tools, okay, that is called in silico. In vitro means we use laboratory biochemical assays, okay, proteomic assays, that is called in vitro. When we say in vivo it means using animals. Okay, so there are three terms: in silico, that is the term here which talks using computational tools, okay, in vitro is laboratory studies, and then in animal studies is always called in vivo. Okay, so there are three types of terminologies which we use. So the in silico takes 2 to 3 years, may cost about 10 million, then we go to testing on animals, that is the in vivo. In between you will also have the in vitro also, that may take another 1 year or more, that may cost $10 million. Then we go through the various human volunteer trials, okay, phase 1, phase 2, phase 3. Again you can see 1 year, maybe 3 years, and these are the cost factors. Okay, so finally it may add up to almost $1 billion US dollars. That is the current cost of manufacturing, current cost of introducing a new molecule into the world. So it is a very expensive process. This is taken from this particular reference here. So as you can see, synthesis, extraction, if you are a chemist you may be synthesizing new molecule, if you are using natural products you may be extracting, and then we are going to test it against maybe anti-cancer drug or anti-inflammatory drug or diabetic related. So biological testing, then we have toxicological studies, pharmaceutical dosage, formulations, then comes the clinical trial, okay, clinical evaluation, that is on the human, and then phase 4 trials, process development, because you need to manufacture in large quantity, then we need to get approval from the regulatory authority, looking at bioavailability and so on actually. So if you look at these cost factors, clinical trials contributes to 25% and so on actually, okay. Then you are going to regulatory authorities, and that also will going to cost a lot of money, biological screening about 12%. So it is an expensive process. That is why when a drug is introduced, a new drug is introduced into the market, it will be very expensive, because cost of introducing a new drug from the lab is a very expensive process, okay. So if you look over a period of time, the cost keeps increasing, the R&D cost keeps increasing, but the new chemical entities are sort of decreasing. That is because the FDA has become more stringent, they want more information, they want to know what are the side effects, they want to know the long term toxic effects of the chemical, the toxic effect of the metabolized product. So that is why the cost keeps going up, and many compounds do not succeed and cross the FDA barrier, okay. And that is a big problem nowadays in drug discovery, and that is a big challenge. So this is a very interesting slide. So it may cost about 1 billion or even 1.2 billion US dollars to launch one drug molecule in the market as a new drug I am talking about. If it is already old and you want to use that particular drug for some other disease, that is called repurposing. For example, aspirin. Aspirin was originally introduced for fever, then pain, later on aspirin is being used quite a lot for blood thinning purposes, okay. That is called a repurposing, that will not cost you much. So it takes about 12 years, 15 years for one new drug. So it is a very long process, okay, as you can see, very long process. We start with some computational studies here, okay, that is the computational biology studies. We decide on the disease, what type of disease you are looking at. Are you looking at inflammation, and are you looking at cancer of certain colorectal cancer or breast cancer. So which target I am going to look at, okay. So I need to identify which targets, because there could be many enzymes, proteins, molecules may are involved, and your drug may be working on only one particular target, okay. So you will try to identify which target. Once you have identified the target, you start designing molecules, and that is what is called a lead identification, okay. Once you have decided, you go for lead identification. So there are a lot of techniques involved. If you want to look at the disease mechanism, how does say inflammation progresses, starting from the site of inflammation right down to various leukotrienes or prostaglandins. So what are the various enzymes involved, how is the metabolic pathway. So I can use computational biology type of approach, draw a big network of various pathways, and then I can see how the flux flows. So that is called computational biology. If I want to identify the protein, okay, if I want to identify the protein, then I may use proteomics tools, we will talk little bit on proteomics, but I will not talk too much on that, trying to identify the protein structure, 3-dimensional structure, its function, its active site, that is all called proteomics. We may have to use some bioinformatics tools because we may have to compare the protein which you have isolated for your disease. Are there similar proteins available, the databases, what are the properties of those proteins, so I can connect with the new unknown protein, okay, that is called bioinformatics. Then the actual discovery of the lead, which involves molecular modeling, quantitative structure activity relationship. QSAR means quantitative structure activity relationship. We are going to talk quite a lot about this particular portion. Activity relationship, okay. And then you need to do the wet lab experiments, that is experiment in your lab, okay, that is where you check the biological activity of your compound. You may use bacteria, you may use virus, you may use animal cells, whatever is your biological assessment. Simultaneously I need to understand the properties of the compound, the lead. Drug likeness, does it have the drug likeness properties? Now that means after all it is going to be consumed by human, so it should not have certain problems, or it should have certain properties like good solubility, good absorption, okay, that is called drug likeness property. Does it have toxicity, short term toxicity, long term? How is its ADME? See, we keep on introducing this term ADME, it is going to come quite often now. Does it have good ADME properties? So simultaneously when we are looking at possible candidates and testing it out in the wet lab biological assays, I need to understand these things also. This is very, very important, because many drugs may have good activity, but they may be very toxic. Many leads may have very good activity, but in the stomach it gets degraded, because as you know the stomach pH is extremely acidic, pH of 2. It may have good activity, maybe it is toxic, or it may have good activity, maybe it gets metabolized inside by the liver and other enzymes involved. So the active drug concentration may be very, very less at the target site. So the ADME could be very poor, the absorption could be poor, the distribution inside could be very poor, or it gets metabolized, so concentration is very poor, or it does not get excreted from the body, so it keeps staying inside, it gets accumulated, and over a long period of time the concentration may be very high, which may be toxic. For example, if I am using nanoparticles, metal nanoparticles, they may stay inside the body, get absorbed by the tissues because they are nano size, and they may have metal toxicity. That is why now use of metal nanoparticles in the body, there is a lot of worry because of this particular problem. So simultaneously when we look at a lead, an active molecule, we may have to simultaneously look at all these. I may have to perform experiments to determine all these parameters, okay, or I can use computational tools to predict some of the drug likeness property, toxicity, ADME, and so on actually. So the course is predominantly going to cover this molecular modeling QSAR. Course is going to cover predominantly some of these computational approaches for determining ADME and drug likeness property, okay. So of course simultaneously one has to see how to manufacture the drug in large quantities, that is called process development, bioprocess. It should have the good manufacturing practices approved by FDA and so on. Once a lead is identified, it goes through the animal preclinical trials, then clinical trial 1, 2, 3, and then FDA approval, and finally gets launched, okay. So this is the pipeline for new drug discovery. So here we are talking quite a lot about computational approaches, computational biology, proteomics, bioinformatics, here we have the molecular modeling, and then here we have the drug likeness, ADME prediction. Okay, so the course is going to cover these two circles, how computers can be used for calculating some of these parameters. That is what we are going to talk about. Okay. So, the drug discovery development process, we can call it the discovery stage, the development stage, the registration stage. Okay. Discovery, like I said, target identification, validation. We need to develop the assay. For example, if I am going to study how my drug is going to bind to a protein or enzyme and inhibit, I should have a biochemical assay. Okay. Maybe a fluorescence assay or a colorimetry assay. Okay. So, I need to, or a radioactive assay, or I may use a animal cell. I may be looking at some metabolites that are produced. So, assay development is also very, very important. Then once I identify my lead, I need to optimize the lead. That means it is a balance between activity vis-a-vis its properties. Okay. So, that is called lead optimization. Okay. Then the pre-development. Of course, the development stage we have the animal studies and clinical studies, and then of course registration, regulatory approval, looking at the life cycle. Sometimes drugs after being introduced into the market may be withdrawn because certain things which have not been thought of, it may be having problems actually. That is why phase 4 is like getting feedback from the people who have been using the drug globally, okay, or in particular continent, and then see if there are any other problems which has not been thought of. Okay. For example, there are many drugs. You have the anti-inflammatory selective COX-2 inhibitor which was introduced into the market, and then it was withdrawn because it had cardiovascular issues on some of those patients who took that. Okay. So, phase 4 is also very important, which is like a follow up. Okay. But as I said, our focus is more on this side of it and nothing to do on that side. Okay. So, but still we need to understand little bit on phase 1, phase 2, phase 3 and phase 4. So, there are 4 different phases of operation. Phase 1 involves human pharmacology, phase 2 involves therapeutic exploratory, phase 3 involves therapeutic confirmatory, phase 4 like I said post marketing, after it has been marketed. In the phase 1 we are looking at any side effects that may be caused by the drug when it is given to healthy volunteers. Phase 2 is looking at what is the dose response. Okay. If I give 1 milligram, what is the response? For example, it is a bacterial like an antibiotic. If I give 1 milligram, how much bacteria is killed? If I give 2 milligram, how much bacteria is killed? So, that is called therapeutic exploratory. Then in phase 3 we are looking at long term side effects. I mean, that is why it is done for 3 years and so on actually. Okay. So, phase 1 we are looking at tolerance, we are looking at pharmacokinetics and pharmacodynamics. Okay, that is a very important thing that happens. Pharmacokinetic is when the drug is given to a patient, it gets a little bit absorbed, then it gets excreted. So, in how many hours does it get excreted? What is the maximum concentration of the drug in the body? Okay, because of the metabolism and absorption. So, that sort of parameters are determined in pharmacokinetics. Okay, so you may take samples either from the blood or target side and see what is the concentration of the drug and so on. In pharmacodynamics, what the drug does to the patient or the target. So, if the drug is there, does the bacteria go down 10%, does it go down 20%, or if you are looking at a tumor, is the tumor size goes down as a function of drug concentration. So, pharmacokinetic is what the body does to the drug, pharmacodynamics is what the drug does to the body. Okay. You also look at drug metabolism and drug interaction and also estimate the activity. In phase 2 you look at where the drug goes and acts. So, you try to look at whether there is an upregulation or downregulation of the target, what is the dosage required to kill a particular amount of bacteria or reduce the activity of some proteins. Okay. So, we develop something called dose response curve. So, if this is concentration of drug, what is the response? So, initially for some concentration there will not be much response, drug fever goes down by 1 degree Fahrenheit. If I give 2 milligram, fever goes down by 5 degree Fahrenheit. So, that is called a dose response curve. Okay. That is what you measure in the phase 2. This also helps you to identify the end points. Okay. When do you stop giving the drug? So, do I stop when the temperature comes down to some certain value? Do I stop for an infection when the amount of infection or bacteria is less than 5000, like that, that is called the end points. Okay. So, all these are measured in phase 2. Then the phase 3 we are looking at safety profiles. That is a very important point in phase 3, the safety profiles. Look at the benefit versus risk, because there are anti-cancer drugs which could be very toxic to the healthy cells, but at the same time they may be killing the cancerous cells. So, we look at the benefit versus risk. Okay. If there is a terminally ill patient and the patient will not survive more than 6 months, so by giving the drug which may be toxic, patient may survive for 2 years. So, is it worth it? So, that sort of studies we do actually. Okay. We do all these things in phase 3. Then of course, phase 4 is after it has been marketed, is there are any other adverse reactions, do I have to again give some warnings to people who are taking the drug. After all, in the phase 1, phase 2, phase 3 trials, the drug is tested only for about 1000 volunteers, but then when it is given to public at large, there could be some new reactions which has not been thought of. Okay. So, that is done in phase 4 trials. Okay. So, drugs fail, lot of drugs fail, very little lead compounds get converted as a new drug enters the market, lot of drugs fail. Okay. Why do they fail? Okay. As you can see, 9 out of every 10 new drugs fail in clinical testing. Okay. 9 out of 10, that means the success is 10% or even less. A drug in phase 3 testing has 32% chance of failure. That means it is crossed phase 1, it is crossed phase 2, but still it can fail because it has crossed phase 1 and phase 2. The percentage failure rate is much lower when compared to this rate. Here the success is only 10% or 90% failure. Once it has crossed phase 3, success the failure percentage is only 30%, because at each phase the pharma company spends millions of dollars. So, if the drug fails and it has to be withdrawn, they have lost a lot of money. So, only 20% of drugs that enter phase 1 make it to the market, 20%. That means remaining 80% fail. So, whatever money they have spent goes down the drain. So, that is why computational tools are being widely used, so that we try to find out possible failure compounds and do not take it further. Phase 1 50% fail, 50%, phase 2 30% and phase 3 25 to 50%. So, overall success rate is 3 to 8%. So, like I said here 10%. Okay. And the percentage of drugs failed for neurological diseases is higher, okay, because neurological disease are much complicated. The drug has to enter the brain region and then do its job, whereas remaining drugs like inflammation or cancer or stomach pain or fever, they do not have to go to the brain region. Okay. The requirements are very different. So, that is why percentage of drugs failed for neurological disease is higher. 200 candidates failed for Alzheimer's disease in clinical testing. Okay. Alzheimer's disease is more to do with the loss of memory and so on actually. Okay. But they have been tested in rats or mice, that means preclinical trials they work, but when it comes to human volunteer trial it fails. Okay. So, of course, there is a difference between the system of rats, mice versus human, but still rats and mice is used widely in preclinical trials because they are the closest to human volunteers and they are much cheaper. Okay. So, many drugs stop working when tested in people. Okay, why is that? Maybe toxicity, maybe when it was tested in animal, slight elevation of some liver enzyme which was not taken very seriously, but when it was given to human, the enzyme level may be getting elevated too much. Even if there is one random fluctuation in hundreds of tests, then they decide that that drug is not very safe and it has to be withdrawn. Okay. 1 in 100, okay. That is very tough business. Poor biopharmaceutical properties, like I mentioned, maybe solubility is very poor, maybe absorption is poor, maybe metabolism is happening either in the stomach or in the liver region because of various enzymes present. Okay, it is not getting excreted properly, or it gets excreted very fast. It is not it is getting distributed too much, and that means the concentration of the drug in the blood is so low. All these 39%. Lack of efficacy, that means the concentration at the target site is not sufficient. Okay. Imagine I have a pain in my finger and I take a drug called ibuprofen. I am sure all of us would have taken ibuprofen. Okay, so the drug is taken orally, so we have the GI, that is the gastrointestinal, then it gets absorbed into the blood. Okay. Then you also have this liver which keeps degrading this. Okay, then which is wasted, and then finally it reaches the target. If it does not get absorbed properly from the GI, again it gets wasted, okay, to the fecus. If the liver degrades this, it comes out through the urine, again it is waste. So finally it reaches the target. Okay. So the concentration of the drug in the target is much less than the concentration of the drug taken by us orally. Okay. So it might not be sufficient to perform its duty. For example, if I am looking at an antibacterial drug, the concentration of the drug should be sufficiently larger than the minimum inhibitory concentration, okay, of the bacteria, that is called MIC. So if the concentration of the drug at the target site is very low, that drug is of no use, that is called lack of efficacy. Okay, understand. Then toxicity, the drug has toxicity, or the metabolites, the drug gets degraded in the liver, maybe those metabolites are toxic, short term toxicity, long term toxicity. So 21%. So you see, if you add all these 39, 29, 21, it is a huge number. Okay. And so the properties of the drug is very important. Okay. The ADME properties, the efficacy, as we can see, which I explained here, because of lack of efficacy, then of course toxicity, many drugs fail during the development process, either preclinical or clinical trials actually. So one need to put in lot of focus in this area, not only just looking at the activity in my lab, anti-cancer activity, it is showing very good activity against certain cell lines, but I need to also understand the physicochemical properties of the molecule also, so that I can design a molecule which satisfies all these conditions in addition to good activity. So that is very important. So a drug is not only doing very well in the lab because it is showing very good activity, but it should also have all the properties, okay, the biopharmaceutical properties, and also this good efficacy, so that it passes the clinical trials and it gets approved. Okay, we are going to that is why we are going to spend a lot of time on these aspects as well, and not only just looking at a candidate which shows very good activity or which has very good inhibitory power against an enzyme or a protein and so on. And of course toxicity is another big issue. One needs to do a lot of experimental studies on fish, on animals and so on to identify toxicity, and there are different computational tools also which can help you to predict toxicity of compounds, the metabolites and so on. Okay. So in the next few weeks we are going to talk more about this before we actually jump into the predicting the activity of various lead molecules. Okay. So we will talk more in the next class. Thank you very much for your time.